In this session, our guest speaker will be discussing his presentation titled The History of Hybrid Capture at the McDonnell Genome Institute. Bob Fulton is the Director of Technical Development at McDonnell Genome Institute and joined MGI in 1994 and has over 25 years of experience in next-generation sequencing and analysis. Throughout his time at MGI, he has led the production-based targeted sequencing efforts as well as the sequence improvement pipelines responsible for genome sequencing refinement. I'd like to thank Bob for his time in putting together today's presentation. We will now begin today's session.
Hi, my name is Bob Fulton. I'm at the McDonnell Genome Institute at Washington University in St. Louis, and I'm here today to talk about the history of hybrid capture at the McDonnell Genome Institute and provide you a variety of examples of things we've done over the years. So hybrid capture efforts at MGI have been around since the early 2010s. From exome capture, custom capture activities. So exome capture, there's been a variety of flavors over the years with various commercial sources from IDT, Agilent, Nimblegen, Twist, Illumina, to name some. And from those various vendors, various products as well, some small exomes, some large exome target regions. And it's been an interesting evolution over the years where everything started pretty large, actually, very comprehensive, included UTRs, very large spaces, and then pretty quickly, people realized that the sequencing costs a lot of money. So we actually got leaner exomes to enable more cost efficiency when doing an exome. And I think the relevance of the variants discovered in some of that ancillary space, the UTRs and things, they were much more difficult to pin down in terms of function or potential function. So the leaner exome became very popular. But the choice of those exomes over the years, it has really been dictated by various projects as well, and the scientific goals of those projects, where in some cases we wanted a bit larger space to capture more variation from any particular genome. And again, some we just wanted the lean coding space to understand those more functional variants. In terms of custom capture, this was really an evolution. It started as a solid phase, so putting the libraries onto a solid phase array and capturing molecules on that solid phase. But that pretty quickly turned to liquid phase as well, with biotinylated baits and for the target regions, those varied greatly from various projects as well, from genes to regulatory regions, viral insertions, regions under GWAS peaks. So we'd get GWAS studies that showed signal. So to extend those from a sequencing level onto various samples, we'd target those GWAS peaks, and then cDNAs as well. And I'll give some examples as we go through here to outline some of those. And then we did confirmation and extension studies. So what I mean by confirmation is we would get variants discovered from various mechanisms, perhaps from whole genome shotgun or whole exome sequencing, and then we could study those sites in other samples or in the same sample to confirm the variant, for instance, or gain more depth on that variant, or extension studies where we would find a set of variants relevant to a project and then extend those variants on other samples to see if those same areas of variation existed in similar samples. We use that to increase the depth to overcome mosaicism, or the custom capture, I should say. We used it for additional depth to overcome mosaicism, clonality, and contamination issues, as well as to refine the allele frequency estimates and monitor changes in variant allele frequencies. And I'll show an example of that. Then spike-ins. So we used spike-ins quite frequently, and we could do spike-ins on custom sets, or we could use these custom spike-ins on the exome to add additional space. So especially on the exome, we could augment coverage from poorly represented areas of the exome or regions that were important to us. We could target viral insertions, and I've got a slide demonstrating that. Identifying distinguishing SNPs for identity of individuals. Coverage balancing, again, if some regions were poorly represented in a particular capture set, we could spike additional probes in to enhance that. An example is the TERT promoter. So the TERT promoter is GC rich for one, so it didn't always capture or represent very well in sequence. So we added additional probes and targeted that region more specifically. Then various commercial sources that was dictated by the synthesis scale and efficiency of the scientific objectives for these custom capture sets in particular. Some commercial sources were better at small numbers of probes, where other commercial sources were better at broader and more cost-effective at broader capture sets. And key metrics that we've always focused on with capture is on and off target percentages, plexing capabilities, so number of samples that you could do in a single hybridization reaction, and then balance of the capture and the coverage. The better balanced things are, the more precise we could be about coverage targets. Then depth dictated by scientific objectives. So, if we're trying to really refine allele frequencies, the more depth, the better. Where if it's just a discovery exercise looking for a germline SNP, then the coverage requirements aren't as great. And then dual index libraries for sample integrity. This became a very important part as we plexed samples, and it also on the patterned flow cells, where you had the index hopping, and again in pools of multiplexed samples, where in amplification processes you could get crossing over of various molecules. The dual indexes prevented crosstalk or prevented index hopping to complicate what variants went with what samples. The dual index ensured that we had the same molecule. So some examples of research projects and applications over the years. Some of these are ancient, some are some are more recent. But the TCGA project, that was really a key driver project, I would say, for hybrid capture activities. And we did SNP discovery, viral insertion detection, confirmation, and extension studies. So it broadly, the hybrid capture broadly applied. And many of these projects were in the earlier days of sequencing. So nowadays, the price of sequencing has come down considerably, and we sort of take that for granted, where back in these earlier days, the ability to target specific regions of interest was critical because of the economics. So another is a pediatric cancer genome project. That was exome and targeted capture. And again, cancer genomes needed the depth for clonality analyses and rare subclone variant discoveries. Cardiometabolic disease study was about 20,000 exomes. So that was one of the larger exome projects we did in the early days, looking for cardiometabolic disease associated variation. Alzheimer's disease, again, just an exome project. Cleft lip. So this was one of our early hybrid capture projects. This is where we really explored the ability to multiplex, where we had about a 6 megabase custom target site or target sites that were underlying GWAS peaks, and we were able to multiplex 96 samples at a time on a hybrid capture, and we had 6,000 samples. So that multiplexing enabled both the production workflow as well as just efficiency onto the sequencing instruments. Pharmacogenomics research network, we did various, understanding drug metabolism, looking at asthma, rheumatoid arthritis as a couple examples. We actually worked on a broad pharmacogenomic target set as well. Age-related macular degeneration, this was an exome project. The regulome. So the regulome was a custom set that we developed. And this regulome space, it was about the size of an exome. So it nearly doubled the exome space. And it was the regulatory regions within the genome. And what this enabled us to do was to focus really on both the exome and regulatory regions to expand sort of the relevant regions of the genome. This is, again, pre-whole genome days. I would think for a project like this at this point in time, unless you needed significant depth, you would just do whole genome. But in those days, the sequencing was very expensive. Did a pediatric ALL project. This was a broad range of targets, so regulatory regions, genes, GWAS targets. So again, broad range of things. Then this AML project, this again was sort of instrumental in our development of hybrid capture techniques. We had what we called the RMG, so it's recurrently mutated genes and those came out of 200 samples from the TCGA project that we had done either whole genome or exome. We came up with a list of 260 genes that were recurrently mutated from those samples. We made a custom capture probe set, and then we utilized that to study further AML samples, and we in some cases would spike that RMG set onto an exome, and what that did is gave us better depth, about a 5-fold increase in depth actually on those genes of interest, while we still capture the exome space. And that the RMG space, actually, it was only about 1.6 megabases. So on a 40-ish megabase exome, didn't have a tremendous impact on the sequence coverage necessary, but gave us very relevant coverage in genes of interest for AML to help tease out the clonality and make sure that we were identifying these critical variants. And then we would use sometimes the RMG and exome together. Sometimes we would use the RMG alone and could get thousands of X coverage from particular samples in that relevant space when we did just the RMG alone. So the impact of depth. So this is, in my opinion, one of the, other than saving on your sequencing dollars, the impact of depth is also nicely illustrated in this slide. This is from Chris Miller. And what you see here are various panes of A through F here of the same 2 genomes. So this is a primary AML sample and the relapse of the same sample. And we looked at various levels of coverage and the impact on the precision of mapping the variant allele frequencies. So in pane A here, we have 26x coverage of the primary and 30x coverage of the relapse. You can see there's clearly shared variation between the primary and the relapse, and then in the green, those are variants that are specific to the primary tumor. And in the red, those are variants that are primarily in the relapse sample. And then the gray there, they're more commonly shared equally between the two. In the next pane, we moved to 52X and 58X. And again, you see similar things, but you see things starting to tighten up in terms of allele frequency and the precision of that. D is 77X and 85, again, tightening. D, we start adding basically subclones to our analysis where you can see additional information. So this is at 186 and 180X coverage. And you see this precision, and you see now in various colors, various subclones from the tumor and the relapse sample. 855X and 310X, you really start to see these specific subclones. And you see and then the next pane, F, this is the most depth that we got. So we were sample limited here. So the 310 on the relapse, it didn't change between those two because we didn't have any more sample to hybridize and sequence any more deeply than we already had. So that number stays the same, but we jump up a bit on the primary. But again, you see these very precise subclones. So the red one, actually, that was ultimately the subclone that grew out in the relapse that killed the patient. So understanding, and you see on the primary axis there, it's only at a 2 to 3% allele frequency. At 30X and 26X, there's no way that you can see that. But at 310X and 1640X, you can actually see that move off the diagonal a little bit, or off the axis a bit, and see that more specifically. So we understand now that that subclone was present in the primary tumor. It was just at a very low frequency. So other things we've done over the years. So this was more of an extension study. So the ATRX gene, there's a deletion that's common in the latter exons of this gene that does have some prognostic indications. So this was part of the St. Jude project, the Pediatric Cancer Genome Project that we did. And this analysis was done many years ago from Charles Liu and Mike Dyer. But this region, this is indicating the deletion. So you see a reduction in depth. And again, this is common in these cases, and it does have prognostic indications. So what we did was developed a set of probes for the ATRX gene. And we actually, so the little red ticks along the bottom there, those are the exons. We added additional probes in the intronic space in these latter exons because this is where the deletions occurred. So in some ways, we were trying to capture the deletion events themselves, but also just to enhance the coverage across there to enable us to see these deletion events. Viral capture. So again, this slide is ancient. This is actually prior to dual indexing. So you do see some contamination levels on this slide. But basically, the take-home message from this is we were able to develop probes to the HPV sequence and several HPV strains, actually. And we developed probes, and then what we did is we spiked those into exomes. So for the TCGA project, for the exome samples that we sequenced, we actually spiked the HPV probes in with the exome, and we were able to capture HPV insertions outside of the exome space. So it still indicated whether these individuals had an HPV insertion and if it was outside of the coding space. Without these probes, only insertions in the coding space captured by the exome product would be detectable, but this enabled us to get the intronic insertions as well. This is a gene fusion that was detected in a particular sample. In the original RNA-seq data, this had like 2 reads of evidence and at substantial depth from the original discovery in the RNA-seq. So we suspected that there was a fusion, but again, 2 reads is never very comfortable. So we developed probes that sat on both sides of the splice site, the putative splice site that we discovered from the RNA-seq, and then also bridging that. I think either one does capture what we want in terms of you don't necessarily have to have the specific splice junction. Just having probes to enhance coverage of these genes was adequate. And what you can see actually, this fusion, the 2 products are on different strands. That's why you see the slope on each side. So one of these flips around and comes together actually when they're, when you put them on the right strands. But this confirmed with great depth this fusion, which was pretty, we were pretty uneasy to confidently call without this. And to do additional, to get this sort of coverage would be nearly impossible from the RNA-seq, and certainly not financially feasible. But with the, any putative fusions like this, we could probe and enhance the coverage very nicely. So current and future applications. So again, current, all the above, and that's just a snapshot of various things that we've done, and I'm always happy to talk about any of those projects and more specifics on them if people are interested. But future applications and things that we are applying hybrid capture to yet today, as I say, all the above, plus single cell and spatial libraries if you're interested in only specific gene sets. And some of these are supplied by commercial vendors, or you can develop your own probe space as well, and we've done some of both. Split pool technologies, very much the same, where again you can enhance specific genes of interest if you'd like. Long read applications, so Oxford Nanopore, PacBio, for instance, complex assembly regions. You can enhance those the coverage from those. Full-length cDNA. Again, those technologies tend to be more limited than short reads in terms of numbers of reads that you get. So by focusing your coverage, that can enhance your scientific discovery power. And then single cell transcripts as well, where again that can be you can capture the full length cDNA from those applications and run those, and you can see the cell barcode and the unique molecular identifier as well as the transcript. So if there are specific full-length transcripts from single-cell data that you're interested in, for instance, expressed variants in the middle of the transcript, you don't get that from the single-cell short-read data very readily, where you can get that from the longer read technologies. And then many other applications as well. So we always creatively use hybrid capture. So with that, I'll end. And again, we've had a rich history of hybrid capture at MGI, and I look forward to continuing that work. Thank you.